Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this session, we're going to explore the concept of gene linkage, and later we're going to apply that to how to do gene mapping. But in this talk, we really want to come to the bottom of this whole idea of linkage, which we've really not discussed before, although you're going to see that we've discussed some things that sort of might have gotten you thinking about this possibility anyway. So I think you'll agree that we have decided that in a normal dihybrid cross, and when I say that, I mean a dihybrid cross is what it is, but if it's of the form of two double heterozygotes, which is how we've been doing it a good amount of the time all along, big A, big B, little b, for example, you guys know that we're going to have a 9 to 3 to 3 to 1 phenotypic ratio if we do the cross that way. And that's because of independent assortment. In fact, remember that 9 to 3 to 3 to 1 ratio was the evidence that in fact independent assortment does take place. Now, over the last couple of sessions, we've been looking at some seeming exceptions, which we realized were really extensions of Mendel's laws. And all of those have led us to a little bit of a deeper understanding, as I say here, of how genes can exert their influence in seemingly different ways without breaking the laws. In other words, they always do this within the context of what Mendel said in his first and second laws. So this really is another seeming exception. We're going to really examine this seeming exception because it's very, very powerful, not only in terms of understanding what's going on, but in terms of actually allowing us to be able to physically map genes onto chromosomes. Remember, there's been this sort of dynamic and dichotomy all along in the sense that Mendel knew about the information in the genes. We then realized that the genes were physically located on chromosomes, and these two concepts are really, really going to be coming together in this lecture or this session. Now, I'm going to give you an actual cross that was done, and you could do it, I guess, if you wanted to, that illustrates what we're talking about here. So using Drosophila, and there's a reason we're using Drosophila. This phenomenon was discovered in the fruit fly, and there's a good reason why it was in a minute. So remember how fly geneticists do this. They tend to name genetic loci based on the mutation that they discovered. So in this case, PR for purple is a mutation. We saw the white-eyed mutation before. Well, this actually causes purple eyes. Okay. PR plus. Now, you could say PR plus or you could just say plus, but that's the wild type. And I'm sure you remember that the normal wild type color for eyes in the fruit fly is red. And as you would expect, you can't always assume it, but it's pretty much always true in these cases, that the wild type plus mutation is dominant to this purple PR mutation. VG is a completely separate non-allelic gene. It's a mutant allele that causes so-called vestigial wings. Vestigial sort of means that they're not very well formed and they certainly don't work. These flies can't fly, which is a bit ironic. VG plus, or just plus, depending on how you want to write it, is the wild type. And what that means is just normal wings that work properly. The normal situation is dominant to the vestigial situation. So what we want to do is set up a cross, as I'm saying, as we always do, meaning that we're going to start with pure breeding lines, but instead of doing how Mendel did when he formulated the law of independent assortment, instead of selfing the F1, we're going to do a test cross, and there's a very definite reason for this. As we're about to see, when you do a test cross, it's going to actually allow you to see what's going on with the gametes in one of the parents. Let's see how that's true. So here's the cross diagrammed out the way we usually do it. These are genes that we're assuming are independent, we're assuming are going to assort independently. So we have A pure breeding line has to be homozygous for PR plus, which means the wild type, which means red eyes, I was going to say this fly has. VG plus is pure breeding for normal type wings, right? This is the parental generation. Then we're going to cross it with, in this case, we would say a double mutant. If we were using pea plants, it would just be the recessive trait. But these mutations are recessive. So little PR means that you have purple eyes, and little VG means you have vestigial wings. So if you do this cross, it shouldn't be too shocking as to what happens. And that is because you'll probably recognize, without doing a big old Punnett square, although you could if you wanted to, that the only gametes that this parent can offer are a PR plus and a VG plus. And the only gametes this parent can offer are little PR, little VG. So when you put them together, you're going to get PR plus PR, a heterozygote at the eye color allele, and VG plus VG, a heterozygote at the wing allele. Now, because of the dominance of the wild type traits, all of the F1 are going to appear phenotypically red-eyed and normal winged, right? The wild type. This is not at all shocking that all the F1 are double heterozygotes, right? We've seen this over and over again. Now, if we were to cross this with itself, you would expect a 9:3 to 3:1 ratio, right? But as I said, we're going to do a test cross instead. So let's see how that would work out. It's actually easier than the 9:3 to 3:1, but you have to adjust your thinking a little bit if that's what you're used to doing. So we're going to take this F1 individual, which is phenotypically red-eyed and normal-winged, but remember it's a double heterozygote, and we're going to test cross it. Don't forget the definition of a test cross. Whether it's one locus or whether it's 2 or 10, you're crossing something that's heterozygous. Well, we framed it originally as crossing something exhibiting the dominant trait with something that's totally recessive so that you could figure out what that genotype was. We know the genotype here, but the reason we're doing a test cross is because this individual here, they only, no matter what, can make gametes like this, right? So since whatever gametes this individual makes are going to be meeting up with this type of a gamete, really what we're doing is we're seeing what gametes are being made over here. Now, if you think of it in the normal way, what gamete types should we be able to expect from this double heterozygote right here? Well, it should be all 4 possible types, right? PR plus VG plus, PR plus VG, PR VG plus, and PR VG. Just as if you were doing the dihybrid cross Punnett square, you'd have all 4 of those on one side. Now, if you were crossing it times itself, you'd have all 4 of those on the other side as well. But since we're doing a test cross, all it can make is that gamete right there. So what you would expect is, look, there's a 1/4 chance that this one will meet up with this, or this will meet up with this, or this, or this. So if you put it all together, you expect, it's a little bit wordy or clumsy, but it's a 1:1 to 1:1 ratio, meaning that 1/4 of the offspring will be each of the possible phenotypic classes based on, really, as you see, while of course this parent is contributing, it's always contributing the same thing. So it's really the other parent, the double heterozygote, that's creating the diversity in this F2, which is down here, right? So wouldn't you expect, maybe this is the best way to do it. From this double homozygote parent, you're always getting this half of the equation here. It's not really an equation, but that's always coming. It's the same in every single one, right? So if the PR plus VG plus meets up with that, you're going to get 1. How does this look? It's going to look red and normal wings, right? However, in this case, you get a PR plus VG, this individual is going to look red-eyed, but it's going to have vestigial wings. By the same logic, this one is going to be purple-eyed and normal wings, right? And this last one will be recessive or mutant at both loci, so it would be purple and vestigial. Now, just take a minute. This shouldn't be that shocking because these are the same phenotypic classes we would have seen had we done it the way to get the 9:3 to 3:1 ratio. In other words, these ones here are parental, right? Because red and normal and purple and vestigial were the same as these original parents. And then we're seeing these two over here, which are recombinant, right? We've used these terms before. They have a combination of traits that are different than that of the original parents or anyone else we've seen in this cross. So that's what we should expect. 1:1 to 1:1 ratio, everything will be fine and normal, except for one thing, as you might have guessed. That isn't what they saw in this particular cross. Another exception, or seeming exception. What did they actually see? Well, it's shocking. Don't get too excited. But they actually did the cross using shorthand for the phenotype. Remember, we can do that. This meaning one that's both wild type at both locations. This being one that's mutant at both locations. Remember, those were both parentals, and it's not a coincidence. Look at the numbers. There were, this was actual data. There were 1,339 of the total wild type, 1,195 of the total double recessive. Both of these, which are the recombinant classes, right? Keep that in mind. There were a lot less of them, only 151 and 154. Now, clearly this is not a 1:1 to 1:1 ratio where you would expect all four of those classes to basically have the same amount, the same numbers. So when they saw this, what the heck is going on? Well, it turns out, as you've probably figured out by now, Mendel's laws don't lie, but something different is going on here. And here's the hypothesis. These particular genes, purple and vestigial, are linked. This is a very important term. And in genetics, it means on the same chromosome. Okay? If you think of the way chromosomes move in meiosis, one whole chromosome goes to one side and one goes to the other side. We've sort of gotten away with not talking about this yet. But if two genes happen to be on the same chromosome, doesn't it make sense that they would move together? Listen, remember why independent assortment actually is true. We mentioned this when we, looked at meiosis and said why they said this is acting just like Mendel's factors or his genes. It's all because chromosomes segregate randomly. And that means genes located on different chromosomes are the ones that undergo independent assortment. It turns out, Mendel didn't know, but all of those traits, those 7 traits that we discussed, they're all on different chromosomes. And I think we've mentioned this before, but pea plants have a total of 50 chromosomes. I mean, n equals 25. So when you pick 7 traits randomly, there's no guarantee, but it's not that far-fetched that they would all be on separate chromosomes. Fruit flies only have n equals 4. I shouldn't have done it exactly this way. N equals 4, in other words, there's 8 total chromosomes. But still, for the same reason we said X linkage would be more likely of genes in Drosophila because there's one X out of the total of 4 haploid chromosomes in a haploid set. By the same token, since there's only 4 in a haploid set, any 2 particular genes have a lot more chance of being located on the same chromosome the fewer of them that there are. So Drosophila, it makes sense. There's a lot of linkage. There's a lot of sex linkage as well in comparison with the pea plant. We've pretty much used those as our model organisms so far. Okay, so I hope that makes sense to you. Now, we're going to definitely examine this in some more detail, but we're going to have a new way of looking at things. If they are linked based on our hypothesis, we're going to be able to redraw the crosses to keep track of and illustrate the linkage relationship. And from now on, we're going to show linked genes in this way because it's a lot easier. All right. So check this out. There's a lot on here, but let's go through it. This is the same cross, but we're now recognizing that these genes are linked on the same chromosome. All right. So in the parental, remember it was pure breeding, but now what we're saying is, okay, look, whatever chromosome pair is, this doesn't really matter. But we're drawing it to show that PR and VG, whatever, pluses in this case, they're on the same chromosome. So we're going to draw a line like this, and we're going to grab it, move it. They're both going to go together. And the same goes for here. So that line represents a chromosome. Over here, they're on the same chromosome. So the way you could think of it, think of meiosis and think about the independent assortment of chromosomes, and you'll see that basically when you do the test cross, what's going to happen? Look how, I don't know if it's simple, but whichever one of these two chromosomes come into the next generation, they're both identical, so it doesn't matter. So one like this is going to end up in all of the F1, right? And one like this is going to end up in all of the F1. Now, be very clear about this. It's so important. If you think of it vertically, you can say, okay, it's PR plus PR. It's a heterozygote at that locus. That's why the term locus is so important, at that location on the chromosome. And it's a heterozygous at this locus too. But both of those loci are on the same chromosome, and that's what this drawing is meant to illustrate. It also means when you do the test cross, right? The test cross parent is the same as that parent up there. So the only chromosome they can donate is one that has both little PR and little VG in there. So look at the F2. There's two possibilities, right? In other words, this is what I'm saying. The reason you're doing the test cross is because the test cross parent only can donate that lower chromosome. That's coming from the test cross parent. So whatever else comes that's going to affect the phenotype is coming from the other parent. And it can either give this chromosome, in which case you'll have PR plus VG plus on one chromosome, or it can give this one. So you'll get two offspring type, right? Again, if you do it the vertical way, this one looks PR plus PR, so it'll have red eyes. This one looks VG plus VG, so it'll have normal wings. This one is the opposite. It's purple and vestigial. So these correspond to the classes with the most offspring. And these are also, which types are these, the parental or the recombinant? These are parental types, and that makes sense because they're coming right from the parents without any strangeness going on. And I just want to go back to the data here to show you this and this. Those are the two types we just got, the ones that are in the highest quantity here. So the question then becomes, where the heck did this recombinant ones come from? Because usually they come from independent assortment. We're now saying independent assortment is not occurring, right? Something's going on. Because otherwise, all of the offspring should look like the parentals, 100% of them. In this data, we see that a lot more of them look like the parentals, but it's not all. It's not 100%. So what could have happened? Well, it has to do with this phenomenon of crossing over, which is now going to become so important. You may recall that when we were discussing meiosis, we said that during prophase 1 of meiosis, that the homologous chromosomes of a pair touch each other and actually exchange segments at that time. That is crossing over. Now we're looking at it in a much more deep way. So remember, homologs do exchange parts at prophase of meiosis 1. And let's look at what would happen if a crossover did occur during gamete formation. And when I say if one occurred, I mean if it occurred between our two genes of interest, purple and vestigial. So I put a black line right here, an X, to illustrate that if a crossover event basically means a cut here and a cut here, and then, if I can draw this properly, a rejoining so that you're going to get that type of chromosome and that type of chromosome. If that happens, all right, what is it going to end up creating? Well, I'm going to redraw it down here, but basically what's going to happen is if you follow this top chromosome along, you're going to get one that's PR plus VG, right? Right like that. And the other one is going to be little PR VG plus. So if a crossover happens, you're going to get these two types of gametes. If you then go ahead and do the test cross, let's follow along and see what happens now. Just like before, the test cross parent can only donate that chromosome there. So this now parent in which a crossover occurred either can donate this to the offspring or that parent can donate that. Now we see how we can get those recombinant offspring, because this one is PR plus PR, which means it has red eyes, but it's vestigial, right? That's a recombinant. This one has purple eyes. This one also has normal wings. Purple and normal and red and vestigial, these are the recombinant classes. These are exactly new combinations that didn't exist before in the parents, and if we go back and look at the data, that's these guys over here, right? PR plus VG, red and vestigial, purple and normal. Those are the recombinants, and those were present in a low frequency, and now we know why. Only these came about when a crossover happened during meiosis between those two loci in the parent. Now, it turns out, we're going to really go into detail about this in the next part, but let's follow the entire cross now and just make sure we understand what occurred. All right? And I'll do a little animation that I hope works. Under this assumption, which turns out to be true, we're starting off with pure breeding lines, right, and recognizing that the PR and VG genes are on the same chromosomes. Now, one thing that's interesting, just say if a crossover occurred in either of these parents, it totally doesn't matter, because there's no way to get anything but PR plus and VG plus, which also, keeps the pure breeding intact. You can rearrange them all you want, but there's only two things there. So it is true that no matter what, this parent will always give you this chromosome into the F1. That parent will always give you that one. You've got your double heterozygote. You're going to do a test cross over here. But now the question is this. So now, if no crossover occurs, you can see that this intact chromosomes here are going to be given to the offspring, and you're going to get the parental types. But I just want to go over one more time. So if that crossover occurs between those two loci, now you can see, right, that's in red there, you're going to get one chromosome that's PR and VG plus, right? And that's going to get donated right down there. If that crossover happens, automatically you have to get the other part of that, which is in blue here, which is PR plus VG, which you can see being donated right down there. And that's how you get the recombinant offspring. Now, what do I mean here? Cytology and genetics. I don't know if we've used the term cytology before, but cytology refers to, well, it technically means study of cells, but it generally means people who are looking in microscopes. And my point is this. This is another key of showing that you can put things together that don't immediately present themselves together. What I mean is, they observed under the microscope these chiasmata. We talked about them. They look like X's when the homologs go together. They didn't really know what was going on there. Well, we now know that those chiasmata are places where recombination is taking place, and we can use that information to put, again, the genetics, the information aspect of it, together with the physical aspect of it. A chromosome literally is breaking and rejoining when crossing over takes place in this situation. So it's always great when things that previously were separate can go together. Now, so I think you get the idea of linkage here, but let's summarize. Genes that are close together on the same chromosome, more about that in a minute, are linked. Independent assortment is not happening anymore. Okay, so If Mendel had seen some of these traits, and who knows, maybe he did and just threw them away. I don't know, because it didn't go along with the idea of independent assortment. So crossing over creates gametes with different combinations of genotypes, and that's how recombinants are made. Now remember, recombinants are made through independent assortment when genes are not linked. If they're linked, the only way to make recombinants is via crossing over. I used to have trouble with this word recombination, and so I'll just say it because I remember always thinking recombination and crossing over were the same thing. It really is not the exact same thing. A recombinant, I've said it enough times, is just a new combination. There are different ways of that happening. Technically, interchromosomal recombination, meaning between different chromosomes, that is what independent assortment is. That always leads to 50% recombination, right? You can go back and think of just a regular 9:3 to 3:1 ratio where total independent assortment was occurring, and you would get 50% recombination if you did a test cross. All right. Intrachromosomal, meaning within the same chromosome, that is what crossing over is, and that leads to less than or equal to 50% recombination. We'll see why that is in a minute, but always. In other words, whether it's through independent assortment or whether it's through crossing over, you can never get more than 50% recombination. Let's check out why that is. So we drew a duplicated chromosome at meiosis 1. It was getting ready to engage in crossing over as this. But it turns out that's a bit of an oversimplification because remember, back to the physical. It's really a tetrad, a four-stranded entity at this time. Remember what this looks like. We'll make two a homologous pair lining up next to each other, right? And remember that we said they're going to intermingle somehow. Well, that dotted line I drew over there is the same dotted line there. So what really you have to realize is that if you want to draw it completely correctly, this right here would have to be drawn like that. In other words, there are really two. These are the sister chromatids, right here and right here. They're identical. These are the other pair of sister chromatids, right? So here's the reason why one crossover, think of it this way. Here's one possible thing that can happen. This can get really complicated. We don't want to get too mind-blowing right now. But here's a situation drawn by the red line that says if this crossing over is an event that occurs between non-sister chromatids, right? That's who wants to trade with each other because the sister chromatids, remember, are identical. So let's just number these. Number 1, 2, 3, and 4. That red line represents a crossing over event between chromatids 2 and 3, which are non-sister chromatids. If that happened, read this down here. If that always occurred, still only 50% of the gametes would be recombinant. Why? Because look, under that scenario, that one there and that one there, they didn't get touched. They weren't involved in the crossover. They were just sitting on the sidelines while the other two were trading their parts. So if that crossover happened 100% of the time, always, you would still only get 50% recombinant gametes. And that's the key of why the range is 0 to 50%. Because I just said if it happened all the time. It doesn't happen all the time. All right? Now, I'm going to go a little into the complexity. I want to erase this right there. Because look what else can happen. Say, it's represented by the blue line there occurred. That could happen. And that, I'll draw in a different color, is non-sister chromatids 1 and 4 having a very similar crossover event. But still, if that happened all the time, the other two are getting left alone, and you're still going to get 50%. Here's another possibility. The bottom line is any two non-sister chromatids can engage in this crossover. But if they are, the other two probably are not. So now we have number 3 and number 1 engaging in a crossover. Okay, but then 2 and 4 are being left alone. So if a single crossover occurs 100% of the time between any two non-sister chromatids in a given meiosis, you're always going to get 50%. But the other thing you have to remember is this. When you're counting offspring like we were before, close to like 3,000 offspring, right? What you're really looking at is really 3,000 gametes, the result of all these different meioses, right? And drawing conclusions based on that. The bottom line is this: you can really go crazy about this, and it depends on your class how crazy they're going to make you go. But it is possible that other types of crossing over can occur, and double and triple and quadruple crossovers. Here's the bottom line. If you sum the probability of all of the things that could possibly happen, you still get a maximum of 50%. And it makes sense because in totally unlinked genes, right, that independently assort, 50% is always the maximum, right, of recombinants that you're going to get. I hope that makes sense. I got to tell you that I remember a time when I thought about this for a long time and didn't like coming to terms with it too much. So all I can say is if that's the problem you're having, you're not alone. But think about this. If it always happened, you'll only get 50% recombinant gametes, and therefore, if you're doing a test cross, 50% recombinant offspring. Now, this also explains, you guys, why the recombinant classes occur in roughly the same numbers. Remember before we had like 153 and 156? I don't really remember, but it was something like that. They're close, and that's because, guys, look, I'll just draw it, it's easier. If a crossover happens right there, well, this chromosome has to be produced into one gamete. This one has to be produced into the other gamete. So they're always going to, in theory, they should exist in exactly equal numbers. But remember, we're getting a sampling of the gametes. We don't know which gamete's going to fertilize which egg. So the offspring are an approximation. We can't count every single meiosis event that ever occurred. I mean, in humans, males make millions of sperm per day. So when there are, without making we're not really ready to go into the human situation, but the point is that when you look at offspring, you're looking at the net sum of a massive amount of meiosis events. Okay? So generally speaking, you'll have about the same number of parentals and about the same number of recombinants. So the important realization that this leads to, that sort of leads into our next topic, is that the amount of crossing over, whether it's 0% or 50% or 7 or 20 or whatever in between, is meaningful. And I know this will make sense to you. The closer together two genes are, the less likely it is for a crossover event to occur between them because they're right on top of each other. The more apart, the further apart they get, the much more likely it is that a crossover event's going to happen right in between them. And this idea allows us to construct very, very important thing, genetic maps. So it's going to be the topic of the next session, but just to recap it here, think about this. If you see recombination happening 50% of the time, that means the genes are pretty far apart in the chromosome, so that lots and lots and lots of crossing over is taking place between them. If you see it happening almost none of the time, they're right on top of each other so that there's just not a chance for them to go together. In fact, it's happened before that the same exact gene, people thought they were different ones, but they never could be split by crossing over, so it was evidence that they really were the same part of the chromosome. But what if it happens 10% of the time, or 20, or 30, or 40? The more often it happens, it tells us something about the location of those two genes with respect to one another on the chromosome. I'll let you think about all that for a while. It's a lot. And I'll see you again real soon at another genetics session.